Okay, good afternoon, everybody. My name is Jonathan Scott. I'm from ESA's European Astronaut Centre in Cologne in Germany, one of the moderators for this session. This session is about health biomarkers, real-time monitoring and disease discovery and diagnosis. So we have 5 different speakers covering a range of different topics who've done work in different research areas and different analogues. So yeah, without further ado, I'd like to invite our first speaker. So Anne-Marie Lepartz is from the Department of Internal Medicine, Rheumatology and Immunology at Friedrich Alexander University in Germany. And I will pass the floor to you.
Thank you. Thanks a lot. So I was very happy this morning to hear that cartilage is also important. So now you're listening to a cartilage talk. And I will basically give you snapshots of the work of the last 20 years already. And if you're interested in more in-depth or more details on the studies, you're happy to read them. So I brought you a picture of healthy cartilage. And if you're after this talk interested in cartilage, inspect your goose or turkey or whatever you eat around Christmas and look at the beautiful joints. So if it's healthy like this, it enables that we can move. And if something goes wrong or if it's injured, then movement becomes very difficult and we decrease physical activity. But we're not very good at monitoring cartilage health. And this is true for the clinic. So if people are developing osteoarthritis, we usually find out very late. And this is a huge problem. For the astronauts, we have no idea really what happens, which is a pity because if we don't monitor something that's deteriorating and then we risk injury for a tissue that we can't repair really, or we need surgery to repair it, we should be able to monitor it. And there was a very recent review on cartilage, on everything we don't know about cartilage, by ESA-HRE, and it just came out this month or so. So if you're interested, that's a good summary. So if we investigate cartilage, we usually use imaging. And our body is really pretty, so it's always super nice to look at pretty MRIs. The difficulty is we will not fly an MRI. And this would have been a question that I would have liked to ask this morning. We can't fly imaging techniques really, or as of now. And then also the resolution is not good enough that we can really detect early changes. So we need something else. And for cartilage metabolism, we have biomarkers. It's very similar to what we do for bone, and they can indicate degradation, synthesis, or just turnover. And the advantage of imaging is always I know what's happening and where it's happening, whereas the biomarkers are systemic, which sometimes poses a problem. But still, I think we need markers, and we need to understand how the markers can predict how the tissue adapts and also how the function changes with that adaptation. So I bring you to the first study that we did a long time ago, and the way I prepared these slides is you will always see roughly what the marker measures on the right next to the measuring tape, and the drug box indicates the countermeasure, and it's across nutrition and exercise. It's not drugs or other drugs, maybe not pharmaceutically made drugs. And we here look at COMP. COMP is matrix protein, cartilage oligomeric matrix protein, and it usually indicates degradation or turnover. So this was the first study where anybody looked at a cartilage marker in bed rest, and also in the slides that will follow, before the dotted line or before a gray box, you always have the baseline. Then in this period, there was bed rest for 14 days and a recovery period of 3 days. In this study, that was the VBR study done at DLR. We looked at control, who did nothing, and vibration training. So you can see very easily that once the participants were in bed rest, the serum concentrations of COMP decreased. They seem to decrease further over bed rest and then recover to baseline. And this was the first study, so we had to confirm. So we then participated in the NOOC study and the MEP study. And these studies were nutrition studies. And they were actually done in preparation of the MNX study, which I will talk about in a minute. And the participants, again, either were just bed rested or bed rest plus a bicarbonate, or bed rest plus a whey protein. Both studies were 3 weeks, and we repeated the same experiment, but what we did in this experiment was that the immobilization or the rest before the blood draws was overnight during the ambulatory periods, and then of course they never get up in bed rest, and again overnight rest in the recovery periods. And I just quickly go back. You see that the decrease here is very steep, and we were a bit worried that if we don't control for movement enough, we would lose that effect. But while the slope is a little bit more flat, maybe, you can still see the immobilization effect. And then in the MAP study, we added other markers, and another one that I want to introduce is MMP3. It also indicates degradation, and it has a very similar pattern than COMP, and we could confirm that in other studies in the future. So then, at that time, one idea was, what if we just have people get up? So to try to find out the minimal activity you need. So in this study, it was a bed rest study, only 5 days. The countermeasure was either just standing up or locomotion replacement training, which was designed in a way that it just replaces what we do in everyday life, and it was not dedicated strength training or things like that. So again, we see the decrease of COMP from baseline into bed rest, and it's super quick. It's as fast as the bone biomarkers. The interventions did not make a difference between the two, for the effect, let's say it that way. And this study was crossover designed, so each participant did all 3 interventions, so we could level out the individual variation between people. And this is a more newer analysis. What we then added to the panel of things we looked at, beyond others, was collagen type 2 excretion. So in contrast to bone, for cartilage, it's type 2 collagen. That's the most important, one of the most important constituents. And this is the excretion. So if something happens to cartilage, if cartilage is degrading in immobilization, we would expect increased degradation and then increased excretion. And this is what we see in that study. And if you focus on the solid lines, which are the means for control group, vibration training, or vibration training plus a nutrition intervention, you can see that they increase, and it's actually, for a lot of them, it's doubling. It's just because of the scatter, it doesn't look as steep. There was no difference between interventions. So to summarize this first bit, we found that cartilage biomarkers respond to immobilization, and that the current countermeasures do not do anything to the concentrations of those markers. And that type 2 collagen excretion is enhanced during bed rest. And I think what is important to understand is that if this is lost, it's gone. And for cartilage is not good at healing or at recovering, not like bone. So if it's gone, it's maybe gone. We don't know. So now I talked a lot about immobilization. Now the question is, can we even measure activity in biomarkers? And one study we participated in was a German centrifuge campaign with the Hyper-G cart project, and the different groups were artificial gravity jumping, sled jumping just without the artificial gravity, normal jumping, and running. And we took blood samples on 5 different time points, and looked at biomarker concentrations. And you can see here that you can very quickly, for these markers, initiate a response in the serum concentration. So between pre and post is only 30 minutes. The participants did the interventions that I just said. It was crossover design, so each person did all interventions. And running results in the highest increase of COMP in the serum, and then followed by the normal jumping, the sled jumping, and the artificial gravity jumping. Other way around, sorry. And we then compared more specifically from the same data set, but a little different analysis, again, COMP, just with between running and jumping. And here you see that the running actually results in a greater increase of serum concentration than the jumping. And the same is true for MMP3. And what I also wanted to show you, this is a different study in a more clinical setting that also looked at COMP and MMP3 concentrations, but in walking, so just a normal half-hour walk. The square is walking at your normal body weight. The diamond is walking with a diving belt, and the triangle is walking in a harness that lifts you up a bit. And you can see here that this also results in differences in the serum concentration. So changing the load is also visible in that biomarker. And why I show this is when we imagine what astronauts are missing or people in bed rest, they're not walking around all day like we do. But this happens all the time. When you walk to the bus station, when you walk to the restaurant, this happens all the time, and also for many other markers that we looked at in that study. So this is the stimulus that is missing all the time for that tissue. Can we relate tissue morphology with biomarkers? And these studies are actually the basis for the idea of the Hyper-G card, for example. So in this study, and it's a little bit older now, but they did the same experiment. They had more time points. So they followed the serum concentrations up to 3.5 hours after walking. And they could see that the kinetics of the markers at 3.5 hours relate to cartilage thickness 5 years later. So there is a relation between the markers and the imaging. We can't really do the 3.5 hours all the time in crew or in in bed rest, which is why we had to adapt and had to adapt the experiment. But yeah, if you go away from just looking at resting concentrations, you can actually see a relation between biomarkers and imaging for these. And then now we're switching to the well-known bone loss gears a bit, but this is also a good example for how we can relate imaging to biomarkers. These are two papers from the T-bone study that I collaborated in with the Canadian group around Steve Boyd. And in that study, we could show that the loss of bone mineral density is related to biomarker concentrations, but it's related to both ways. So CTX for degradation, but also to the formation markers. So in that study, it was the people with a higher bone turnover were those who lost most bone. And now I come to the end, because if you go back to the first slides I showed you, I said comp increase indicates cartilage degradation. In bed rest, it decreases. All is good. So super good for the cartilage. They're healthy. So this is now crew data from the ESA cartilage study, and it's still preliminary. And you see launch -60. The gray box is flight, and this is R+1. So it looks very nice. Nothing happened. They're all happy and healthy. But then we found this. And the values of comp increased dramatically, and this is only percent change, but especially the upper two, they go way beyond what we have as clinical cutoffs. With these values, you would say that person doesn't walk anymore or needs joint replacement or something. So with this, I just want to show that the time of the measurement also is super important, because if we had stopped here or here, we would have said, okay, it's fine. We didn't, and then actually, as a result of this, we added a 1-year time point, and now in other studies, we add longer follow-ups. So to summarize this, cartilage biomarkers are mechanosensitive, and we have to know this and standardize the study conditions. So if we don't have adequate resting periods before we measure, we get very noisy data. But we can maybe use this as we do in stress ECG, for example, and use stressed conditions as an output parameter. Biomarker concentrations can reflect morphological change, but we need to identify sensitive modalities and also the timing of the testing. And we're very lucky right now because we have a lot of things to do. So we have joint cartilage experiments going on in Vivaldi, Brace and Brave, but also have a pre- and post-flight crew experiment and are part of CIPHER. And in those studies, in CIPHER, it's only the first-line biomarkers, but in-flight biomarkers. But in all the other studies, we can combine biomarker concentrations with imaging of cartilage, but also the subchondral bone, and also monitor physical activity to see how that relates. And with that, I thank all the people and funding who's involved, and I'm happy for questions. Questions? Do you want the crew, you mean? The crew data that I showed at the end? The comp data? Mm-hmm.
Circulating concentrations are lowered, and when they are rising, you say, oh, that's good. Well, I do not know whether that's good, but clearly the steady state levels are changing. Doesn't that attest to the dynamic nature of cartilage? And isn't there the hope that regeneration is possible?
I hope I got your question right.
What does the marker actually mark? That is my question, essentially.
Say it again, please.
What does that marker actually mark? What process does it mark?
Yeah, that's a good... So we had that question on Monday already, I think. So for cartilage, one difference to other tissues is you need the walking to get a lot of the metabolites out of the tissue. So I think what happens in bed rest is we see lower concentrations, first of all, because the movement is missing and the movement doesn't squish the metabolites out. When we have longer bed rest, and now we're in the 60-day bed rest, then we see gradual more decrease over time in bed rest. So this then I would suggest is more metabolistic than just the mechanical, the metabolites don't get out. The crew data, I think the one part is that what was accumulated maybe in the tissue is now coming out, which we would be able to confirm that it's at some point ends when we have the 1-year follow-up. And I wanted to say something else and I forgot. I don't think, and then in the clinic, when values are going very high, then that's a sign for, or that's supposed to be a sign for massive cartilage destruction. But I also think that we have to rethink these values, because for healthy people, maybe that's not the case.
Yeah. Well, I do not know. It was an innocent question. Mm-hmm. You could interpret your data differently by when the steady state levels are lower, there is less required because there is a lower turnover. Thanks to lower mobility.
What will be interesting, I think, and we see that in bone now too, that there seems to be something going on with the osteocytes, for example, and it could here also be for the chondrocytes. If they're missing this signal, they don't do anything anymore. And do they die, or do they wake up again, or what is happening? Yeah.
Can I? Just a quick follow-up question. The source of COM, for instance, here you showed a picture in the beginning from the knee. It's most likely the knee, so the joint.
It's it's everywhere in the cartilage, and it's also a little bit in the tendon, but the majority of it comes from all articular cartilage that you have. So in any joint, it's just that we focus on the knee because it's from the imaging perspective, that's the best site because it's where it's the thickest and most researched. So we relate it to the knee, but of course.
But is there a correlation with the body weight? When you have more load on the knee when you're back, so this should be maybe the cohort.
Yes, I mean, you saw in this one study I showed where we added load. In a healthy person, if you add load, the slope of the increase that you measure in serum in response to walking is greater with more body weight.
But I mean, with the crew,
with the crew, we haven't looked at that
when
yet.
they have the body weight, so this might be interesting.
Thank
you very much.
Thank you, Anne-Marie. Okay, our next presentation comes from Professor Martin Fiedisch. Professor Martin Fiedisch is a professor of experimental medicine and internal biology at the University of Southampton. And he will talk about oxygen, aging, and take us on a journey across this area in a range of different studies, if I'm right.
Thank you very much, John. And thank you for getting me here. And it's a great pleasure. I learned a lot since yesterday. I want to take you on a philosophical journey, if I may. What can you do in 10 minutes? I'll show you some data, so it's not all made up. But I'm interested in what keeps us healthy. Yeah, the human body is a complex adaptive system with many component parts. We know lots about the component parts, less so about how they interact. And each of these parts are important, but none of them are significant to provide resilience. What is it that provides resilience when you're young? How do I forward using this, right? Thank you. When I was a kid, I was glued to the TV. So for the Star Trek fans, the tricorder was the device Dr. McCoy was carrying with him, and had a nifty little device that was detachable, and when you pointed at your fellow travelers or at an alien or at a rock, it would tell you exactly the flux rates of arginine and oxygen and the elemental composition, and it gave you not only the diagnosis but what to do in this case. Fantastic. I contacted Hollywood. They ran out of prototypes, so we have to do it ourselves. What do we know about health and resilience? Well, surprisingly little. And we know it's an active process that keeps us healthy if things go right. But we don't know what happens during the transition to ill health. The reason why we know so little is how we practice medicine today. And typically patients go to the doctor when they are not feeling well. And the current focus is clearly on disease and not on health. It's not health care, it's care of the sick. Yeah? And despite the promise of personalized medicine and network medicine and all the rest of it. So it also has to do with how research is rewarded and how it's funded. And in so doing, we know more and more about less and less, but we lose spatio-temporal context. And we do not study often the interrelationships. That's significant if we want to, or you as a flight surgeon want to give advice what to do to stay healthy. It's not immediately straightforward. How do we figure it out if we don't even know what's happening on Earth? Well, we need clearly local capability to monitor lots of things. What are the biomarkers most advisable to monitor? I do not know. What process do they mark? We just discussed it in the previous one. I want to leave some take-home messages before we rush in the end. There are important lessons to learn from a range of disciplines that are maybe not immediately obvious. Some are, but complexity science is one of them. Radiation biology we discussed earlier. Nutrition is another one. I strongly feel we need a conceptual framework to approach this. What that looks like, I do not know yet, but it would be a useful exercise to reorganize the literature according to that framework. Nutrition, I think, is absolutely fundamental for this. That's the foundation for resilience. And it's very important to have the technology to get us there, but likewise it will be important to provide funding to do the fundamental science, and we discussed that earlier. One of the colleagues from the DLR had the question, how do we make that happen about human health and not disease? Yeah. So why is everything, I get that question often, that's why I put it up, everything in life linked to redox processes? Well, that is relatively straightforward because life did not develop in isolation. It always co-evolved with its environment. It's always connected to its environment. And whether it's a microbe or a fledgling plant or a seed germinating, or you're cutting a deal with your next best competitor. This is an interaction and a negotiation with the environment. Okay? And that environment has changed a lot in the first 2 billion years of Earth's existence, and that's why, that's, I think, the reason for redox biology being a significant player in this field. Sensing to the changes to the environment has always been and continues to be essential for survival. So what does that mean for space physiology? We need to get that environment right, yeah? We have developed a conceptual framework on the small species interacting with nutritional elements here. I don't have time to go into the details. We call this a reactive species interactome. But the take-home from that is the circulation is the communication highway for all of this. It's not just a convenient way to help yourself to biomarkers to gauge the state of your patient or your astronaut. It's the inter-organ communication highway that connects all cells. Okay? We heard a lot about radiation throughout. It boils down to whatever the stresses are: mitochondrial dysfunction, oxidative stress. Oxidative stress is everywhere, but it's not just the oxygen that is the problem, okay? The reactive nitrogen, reactive carbon, reactive sulfur species interacting, and that then translates into physical and mental exhaustion, bone and muscle loss, cartilage loss, we heard just about. And that's true for astronauts, that's true for mice being transported to the International Space Station. These are 3 papers from Masa Yamamoto's lab in Japan. The resilience and the inter-individual variability, that's what I would like to convince you, are likely explained by the differences in the ability of a body to achieve redox balance. Now, what is that? Well, I don't know, but I know what it's not, okay? This is what it's not. This 20 years have misled us into thinking, oh, you just have too many oxidants, just put some antioxidants on the other end and the body will sort it out. No, it interferes with signaling and it makes matters worse. Okay, it's not that simple, it's slightly more complicated. Well, is not helped that we on Earth we have an oxygen-centric view, which is surprising in a way. Yes, clearly oxygen is required for aerobic organisms, but 7/8 of the development of life on Earth occurred in its absence. Yeah, please notice the axis break here. Oxygen came onto the Earth relatively late as a pollution from aerobic respiration. First, very little was happening because there was a lot of iron and sulfur buffering, and only after the great oxidation event it occurred, when all of the iron was oxidized in the sea, in the water, it arose in higher concentrations in the atmosphere. Okay? We came on here pretty late in the game, yeah? So that means, if you think this through, everything that's required for metabolic flexibility and resilience happened long before, and it happened in a sulfur-rich world in the absence of oxygen, okay? Oxygen is the stressor. We have dealt with reasonably well. We make the most of it with the help of our mitochondria, but the mitochondria, sorry if I can flip back, whoopsie. They originated here, were a product of the incorporation of alpha proteobacteria into archaea. Archaea were very happy. They are threatened by the presence of oxygen, but they do very well in metabolizing sulfur species. Okay, you also need nitric oxide, and we spoke about the ROS, and I will get back to that in a minute. This is all published stuff. When we talk about nutrition, it's not about just the building blocks, it's also the inter-organ exchange of building blocks. Yeah, so we need to get the environment right, and we need to get nutrition right, I think. We try to stab at this interesting conundrum using a variety of cohorts. Here are some of them. This is a perioperative hyperoxic one. These are two hypoxia cohorts. There is a centenarian cohort to consider aging-related changes, but I will show you something from the Marcy cohort, where we have been lucky that ESA supported the infrastructure So we are looking at the continuum from hypoxia to hyperoxia, across the age range. These are just if questions later come to the cohorts. This is the ascent profile from the cultural extreme Everest, going up Everest base camp. This is a chamber study simulating the conditions, asking a question: do you come back from high altitude or from a hypoxic episode a different person? And the question is, yes, you do. So what we learned from this, lots of things, but one important one: fitness at sea level does not predict how well you do at altitude. That's meanwhile old news, but a sojourn to high altitude leaves an epigenetic imprint of that experience, and a metabolic rewiring you will not be able to erase quickly. And this is just a reminder for me to tell you that enhanced NO production is very, very important, whether you look at lowlanders ascending to high altitude or people living at high altitude for extended periods of time. And NO is protecting the umbrella to protect the sulfur circuit from over-oxidation. Okay? Oops. This is now 3 more slides from the Marcy study. Very small number of participants due to the size of the facility. We took lots of blood, saliva, and urine to the level of the participants' approval. We measured a number of physiological markers, very simple things, after a simple exercise test, and they had fun. This is what happens for 3 months. As you probably know, during the summer, sun doesn't rise anymore, and they're very, very happy when they see it again. So there are simple things we can learn. One I want to point out is the variability in responses. So this is just looking at hand grip strength. Okay, we were talking about muscle mass. This is muscle quality, not muscle mass. You see there's a big variability over that year of observation. It's very simple. It's clinically meaningful. And I think it's an integrator not only of your capability of pulling weight with your hand, but also of your mental state. And you see that this goes up in some, it doesn't change in some others, it goes down in others. Enormous variability. You can say, yeah, sure, these are different folks. These were all healthy young male individuals. We had a nutritional intervention, but for sake of time, I skip this to the next. What happens to muscle mass? They were of a very different constitution that has a similar variability, but nothing happens during the nutritional intervention. Okay, but there is clearly a change, and you see this in tabular form here. The biggest change we see is not in muscle mass, it's actually in fat mass. And it's over that year, it was about almost 40% on average in everybody. That's a lot, yeah? That is, this dominates the heat map here. If you take that out, then you see the variability in all of these other markers from the bioimpedance measurement we have taken. So there is a lot of difference, and you see that some do not change at all. Some gain muscle mass, for example. Some lose muscle mass. It's very, very interesting, and making a direct comparison really difficult. That's my last slide, promise. So we need to consider nature, intensity, and duration. We discussed duration of exposure earlier today, I believe, or yesterday. So most of the information we have available relates to short-term space travel, right? Longer duration missions can exhaust reserves, and may compromise the ability to cope with other stressors. In that context, I think of non-essential amino acids, like when we just heard cartilage, the immediate first vote is glycine. We can think of glycine availability, seemingly a non-essential amino acid. Does anybody worry about that? Probably not. Do we have glycine tablets on board? Probably not. Do we need to worry? I don't know. But the ability to cope during longer-term mission will depend on getting nutrition and environment right. And with that, happy to take any questions if we have time.
So some time, some quick questions. Anyone?
So hi, triggered by your images of Antarctica, is there any lesson to be learned in the direction you're speaking about from, for example, expeditions of about a century ago of people traveling to Antarctica for over a year or 2 years?
Absolutely. Yeah, there's lots to be learned, but we should perhaps be cautious in how to interpret those results. We all do, if we do hypothesis-driven research, experiments for a purpose, interpret this one way or another. I think it's time to develop an overarching concept and reorganize the information that is available, as I said earlier, in attempts to maybe paint a more holistic picture, like Alexander suggested in the earlier panel discussion. We need to look at the whole individual. But each of them, medicine is a collection of N of ones, yeah? So we have a tendency of, say, what's your blood pressure? Is that normal, within the range of normality? Fine, you're good, compared to your age. But that started from the time of tables of life expectancies of insurance companies, yeah? That gives you an idea about the bigger picture, yes, but that's not the whole study. Some people may have slightly elevated pressure, slightly elevated cholesterol levels. They will be fine. They will be reaching 100 without a problem. What can we learn? I think we need to rethink our definitions of essentiality of building blocks. That's why I brought up the example of glycine. Coming back to cartilage, glycine is you need a lot of glycine to make cartilage, yeah? But also to make heme. You want to transport oxygen? Well, you need to make enough heme, not just iron. What is the turnover rate of what? How much do we eat glycine-wise? About 3 to 4 grams per day. I do not know what that is in astronaut quantities, but roughly that. What is the daily requirement for glycine, total body turnover? 15 times that. Where does it come from? Nobody knows. Does anybody worry? No, because nutrition is not very fashionable anymore. Might this be a considering factor during long-term missions? Maybe. Might this be an area worth investigating? Maybe. So there's lots to learn, but I'd just throw it all up. This is just one example, yeah? Alex?
One remark, I liked very much the big picture that you has displayed on how we adjusted also to the environmental changes and nature and in the kingdom of animals when the oxygen was absent, not there, and then it rose up to 23, I think, in the Cambrian explosion when all the plants were there. And you have highlighted as well the hypoxic part, which I agree. I just want to add that there's maybe we learned in these 500 million years to cope with the 20% oxygen with the epigenetics and everything. But we have not learned at all to cope with hyperoxygenation at all. There is no concept in our system. So when we deal with high oxygen concentration, the clinics, for instance, no one really knows what is good and to which extent we need the safety margin. When you think about astronauts going in EVA,
absolutely,
rebreathing and so on, no one really knows about these effects because apparently this was never happening in our history. 23% was presumably the highest.
35% was a maximum. Yeah.
Well, it's still a debate. I don't know.
Humans were not around.
We're not around there. Yeah, exactly.
Yeah, exactly. Yeah. Absolutely, yeah, and this is why there's currently a very big discussion in anesthesiology about the best level of oxygen being given during surgery, and the WHO recommends that every patient should get 80%. What can go wrong with oxygen, right? Comes out of a wall. Well, good luck. We looked at arterial and venous concentrations, and the body shuts down in terms of oxygen consumption at the tissue level with 80%. Is that to worry about? Maybe not when this is a 12-hour surgery, but if this is an extravehicular activity that is recurring during lunar habitation, then that may be a very significant factor. We are flying blind, right? I don't know.
Yeah. So yeah, one thing also to keep in mind, of course, is it's the partial pressure, right? So we're operating in a much lower pressure environment when we go on EVA. So we really do need higher oxygen concentrations. Otherwise, it will not be compatible with like. Yeah, exactly. The titration is very hard. And I think it's also important, especially if we have long duration exposures. Yeah. So I totally agree with that.
Yeah, thank you.
Could we save the questions till the end if we have time, if that's okay? Yeah. Super. Thanks, Mr. Rees. Thank you. Okay, next up we have Professor Damien Bailey. Professor Bailey is the Professor of Physiology and Biochemistry, Director of the Neurovascular Research Laboratory at the University of South Wales, and he will talk to us about redox regulation and neurovascular unit functions.
Good to see you. Thanks very much. Thanks very much, John. Good to see everyone. Professor Feelish talked about individual variability, so I'm going to take a very... You've given me a wonderful backdrop to my presentation. I'm going to talk about a dedicated study. I thought it would be really useful for the early career researchers, of which there are many, perhaps more middle career researchers to later career researchers, in terms of some of the hotspots with conducting some of these experiments, and perhaps as well a sense of adventure to come out of the presentation, that the science is actually really good fun. Ask some really ambitious questions and maybe you'll be able to pull it off, but there's some really interesting science that you can do. So I'm going to focus on relativity, which is a Concordia-based study rather than give an overall backdrop to the science that we're doing. So at the moment, we've got an eye on a very interesting planet. And as a physiologist with microgravity and isolation and confinement, it kind of gives me this sense at times, but it's a great challenge. It's an integrative challenge, and it's a challenge that we really must step up to the plate to solve. So over the years, too many to mention, my laboratory, we've been interested in oxygen as a fundamental molecule and gravity as well. And as Professor Feelish mentioned, these are wonderful stimuli to stress the system, to test resilience, and to see how adaptive we are as a species. Sometimes it can go wrong, and we like to push the system very hard. The human brain is an obligate neurobe. We've evolved brains that are disproportionately large. They're about 16 times more expensive to run as far as oxygen and glucose consumption is concerned relative to skeletal muscle. Your brains right now are consuming around about 25 to 30% of your entire basal oxygen energy budget. But yet it only weighs a meager 1/50th of your body mass. If you're lucky, about 1.4 kilos. Mine is probably a little bit smaller. Say no more. No relationship between intelligence and size of brain. So, just get that one out there. So the space exposome, this is a really challenging environment. And as a physiologist who's interested in environmental extremes, it doesn't get any more extreme than space. Of course we're protected with the spacesuit. But the isolation, the microgravity, the radiation, many of these stimuli we don't look at in combination. We look at this in situ, and it's the combination. It becomes exponential in terms of its impact on what I call the space integrome. So, as Professor Feelish mentioned, there are many organ systems that are affected here. And it's really important that we look at this in an integrative, mechanistically translational viewpoint. And of course, this is where the biomarker field has exploded, the wearables, something practical in science that we can deliver on various analogues and certainly in space. And what I'm particularly interested in is the redox proteome, but allied specifically to the brain. Forgive me, the cardiologists and the pulmonologists and the renal physiologists, but I just think the brain is really important. But it's integrated. It's integrated. It's not just standalone. And as we mentioned before from our flight surgeon, of course, it is about partial pressure. And ultimately, at the end of the day, you're operating in a seat which is hypoxic. You've got hypobaric hyperoxia. But the ultimate PO2 is low. And it took a long time to get papers published that hypoxia less oxygen drives an increase in oxygen-centered free radicals. Reviewers really couldn't wrap their head around that. 2019, we had a Nobel Prize for it, and it's a given now. But there's going to be extended explorations, extended EVAs, and exposure to hyperbaric hypoxia. And hypoxia is a little bit like the bête noire. It's not really be considered within the space exposome and its impact on the integrome. Concordia is just a wonderful analog. I've spent my life climbing mountains, doing lab-based studies, trying to deliver experiments in really harsh environments. This is perfect. You've got 12 months of a crew in an isolated environment at 3,500, 4,000 meters. And it's a wonderful, wonderful analog. And those of you early career researchers that haven't looked at this analog, please look at OSIP, please speak to ESA members and access some of these opportunities, because it really is a fantastic, really fantastic analog, White Mars. So of course, our experiments over the years, we've been looking at integrated responses, cerebral blood flow at the top there, trying to define kinetics over prolonged periods of time. And of course, it's very difficult to spend 12 months at Everest Base Camp. The last time we were there for 6 months, and it nearly killed most of us. Very, very hard. This is a typical lab-based experiment. We're clamping the gases here, oxygen from CO2. We don't want CO2 to influence what we're doing with the brain. We want to focus on oxygen. We can place arterial jugular venous lines so we can measure what goes into the brain, what comes out of the brain directly, as you can see there. Synchronous sampling. We're sampling using duplex ultrasound across all of the arteries, extracranial and intracranial, and doing lots of other measurements. So we've spent a lot of time trying to understand how hypoxia impacts the brain, both from a metabolic, molecular, hemodynamic, and also a structural perspective. So this is a friendly chap. He's my 10-year-old son, Luca. He's just going to explain a very complex phenomenon to you, and that is effectively, he understands in my family, even at 10, it's publish or perish. So he's still writing this up for Nature at the moment. So I'm asking the question, Luca, what is a free radical? Ultimately, we talked about redox stress, we talked about reactive oxygen, nitrogen species, but here's a free radical, as you can see here, any atom or molecule that's lacking one or more electrons in the antibonding orbitals. This is for example, vitamin C. It might be a sacrificial antioxidant, and what that radical is going to do is snatch that electron to stabilize itself. All atoms and molecules want that paired electron setup. And over the years, this has had a very bad press because these can rip through the circulation and damage biomembranes and cause damage. Brain damage, of course, is one of the issues that has traditionally been a concern. But as Professor Felix mentioned, we're now slowly understanding that these are acting as redox switches. They're very, very fast signaling agents. A hydroxyl radical, 10 to the minus 9 of a second, that's a billionth of a second. That is a very, very fast transductant. So are they being used in a neuroprotective way? So merci, Luca. He's Welsh and French. So we use electron paramagnetic resonance spectroscopy with a variety of diamagnetic spin traps in order to trap these paramagnetic radicals, and we can identify the species and the concentration of different radicals. And of course, this allows us then to selectively target these radicals. In addition to that, we measure nitric oxide, the principal vasodilator, its bioavailability, which drives vasodilation, getting blood flow to the brain, precious oxygen and glucose, can be inactivated by some of these radicals as well. And in addition to that, of course, we're interested in the redox proteome, shotgun proteomics. We measure just about everything in oxidatively modified proteins with the aim of being able to deliver something that's very specific to the brain. What is a biomarker or a compendium of biomarkers that reflects neurovascular unit integrity or damage to the brain, if you like? So the rationale of this experiment, at high altitude, Concordia, individuals present with debilitating headaches, something we call acute mountain sickness, and also other neurological sequelae, including cognitive impairment. What are the mechanisms that lead to that? What is the role of radicals, or the redox changes, that could contribute to this? And what are the countermeasures that we can deploy to prevent this? So we were interested in an integrated mechanistic approach. We wanted to focus on the neurovascular unit. So this is how the whole integrates with the brain, and the impact of long-term 12-month acclimatization to hypoxia. And we were interested, with the redox proteome, interested in the reactive oxygen nitrogen species and some of the changes that occur to the neurovascular unit. So on the top there's studies that we've performed in the past. We've done diffusion-weighted imaging studies of the brain. This is an example of a healthy brain. Believe it or not, it contains hemosiderin deposits, because in hypoxia, blood flow to the brain increases, it vasodilates, and the red blood cells extravasate into the parenchyma. And that leaves a lifelong, irreversible iron oxide 3-hydroxide metabolic signature in the brain. This is a hemosiderin deposit. So all of your brains, when you go skiing this Christmas, will swell. We're not really clear what that swelling means, but it's something that we need to investigate. That's because of the lack of oxygen. So here you can see we've taken, and I've understood, of course, in space, we always need a wonderful acronym, molecular, hemodynamic, structural, and clinical changes. So we want to take a truly integrated approach of the neurovascular unit in order to understand some of the clinical sequelae that present, again, as a result of this lack of oxygen. So, again, from molecules, the lungs are very important, of course, the heart is very important, it gets the brain to where it wants to be, but ultimately it converges on the cerebral circulation. And this is what we wanted to deliver in Concordia. So multiple measurements that you saw in the laboratory-based study, which is quite a challenge, of course, because it's quite isolated. And in addition to that, we had optical coherence tomography, led by Dr. Susan Mullen, and Alex Sinclair from the University of Birmingham, with the help from Heidelberg. A wonderful opportunity to forge links with industry as well. And in addition to that, we wanted to fly the first ultra-low field portable MRI scanner to Concordia. So this is around about 600 kilos, developed in the US, to allow us to then sequence and do images regularly of the brain over time. So as a workshop, I thought, let's talk just briefly about some of the lessons learned and some of the hotspots, ultimately so that those of you that are thinking of accessing this analog, you've got some idea of what's going on and what to expect. So first of all, huge thank you to the staff. Angelique, absolute legend. Without Angelique, we would never have been able to have achieved most of this. And Malika Balko, who is also responsible for this. We had a research MD, Jessica Studer. She was delivering the measurements. She had to be trained in my lab and then delivered all of these measurements out in Concordia, which is a real challenge. So phase one of any research study in my lab is you take the MD to the pub with your dog and your family, and you strike a chord so that they can understand where you come from and what you're interested in before, of course, we launch into 2 weeks of 20-hour days trying to get them trained up in a variety of different techniques. NVHAB for the baseline, the pre-BDC testing, and this is, by the way, an experiment that's still ongoing. We've only just finished the Concordia winter over at the moment. Samples and people, as we speak, are coming back to Europe. But huge thank you to the staff under the auspices of Edwin Mulder. And again, those of you that have experienced the DLI, this is a phenomenal facility, so incredibly well organized. And again, of course, we get to meet the crew. Ruthlessly well-organized schedules for data collection, fabulous spaces in order to perform the experiments. You've got Peter Zuulenberg here as well, our neuroradiologist, and the team working together, and you can actually have a little bit of fun as well with the crew. But very long days, extremely well-organized. Just make sure that you are as well organized as the DLR crew are as well. That's really important. Equipment transfer. Make sure you've got funding in place. That's really important. And plan your logistics really, really carefully. Expeditioning has taught me that this is the best way to transport material, expensive material long term. Barrels, great, easy to pack. And effectively, we had to get this over to the French Polar Institute in Normandy in France, and a big thank you to Colline Marceau for her work as well. Lots of communications, lots of emails. I think there were about 280 emails between us all before we got to the pre-BDC testing. Huge amount of communication, so beware of that, a lot of work. This is what some of the packing looked like in the lab before we're about to go. And Peter Zuulenberg as well had a specialist MRI crate designed in order to house the hyperfine swoop to get it to Concordia in one piece. Of course, that's in the lap of the gods when it leaves Europe. And in fact, wonderful excuse for Peter to fly with the MRI scanner to Concordia as well. So he's sitting next to the scanner in the basilar plane. Had to be a basilar because at the end of the day, that was the clearance dimensions to get the scanner in and out when it arrived in Concordia. There you can see Peter. And this is the MRI scanner being tracted into the Concordia station, and this is its setup. And again, a practical point for those of you that want to take an MRI scanner to Concordia, the minimum operating temperature is 15 degrees centigrade. And it seems easy to achieve. It's not always easy to achieve in Concordia. Everything grinds to a halt if it's below 15 degrees centigrade. The other thing as well that I was very patently aware of is the certified cost of a scientist of Concordia is roughly equivalent to what a professional Polish footballer earns. It's a lot of money. It's a lot of money. So you need to be well organized, and you want them to be as well trained as possible. So it's really important to make sure you build in enough time. A few challenges and pitfalls. Here you can see Thomas Pesquet, who visited the station. Well, when you're collecting bloods, of course, they're polycythemic. You're hypoxic. You're making lots of red blood cells. When you collect a blood sample and you spin it, you're not going to extract anywhere near as much plasma or serum. So you need to factor that into the equation for the metabolic analyses. Different sampling methods as well that might be a little bit alien to the MD. It's a shame you can't hear this, but we've got a neurovascular coupling technique here on Thomas Pesquet. He's got his eyes closed, he's got his eyes open, looking at how the brain responds to this. So Thomas was working absolutely perfectly here, fine specimen of course, but unfortunately one of the probes broke, and of course the research MD reaches out. There's not a great deal you can do when you're several thousand miles away. So we tried everything we could to fix that with the electronic engineer, but ultimately at great expense we had to fly another probe out. That's something you need to factor into the equation. And the same with the OCT, huge amounts of troubleshooting. WhatsApp saved our lives, back and forth with WhatsApp, trying to get around some of the glitches. And again, with NVHab, under the auspices of Dr. Claudia Stern, very, very friendly, constructive, helpful information to get the very best data that we possibly could. So be patient, understanding, sympathetic, flexible, and available. Data transfer is like a tortoise. You need to factor that into the equation. WhatsApp, all communication really occurred via WhatsApp. So we were running tutorials for Jess, the MD, at Concordia. Some of these techniques are quite difficult. So of course we were doing these techniques with her, which was quite a challenge. Sorry, quality is not that good. I am doing it on myself. So Jess was desperately coming up to speed. We tried everything we could to repair some probes, of course, always a bit of a challenge. But they were light-hearted times. Sunset in Concordia, which coincided with sunrise in Wales, and then Jess, by her own admission, okay, we are in paradise after all. This was the elephant in the room in terms of data transfer. You need to factor that into the equation when looking at files. And also the first couple of weeks for the MD are quite stressful. There's a lot of psychological stress. They tend to be quite ill, infectious illness. But also, you're flying into altitude, so you are going to be suffering with hypoxia-related sequelae. Again, you need to factor that into the equation in terms of how operant they are in terms of collecting data. And of course, Jess is now, I think, in Australia drinking margaritas, decompressing after 12 months of hard work. So, remarkable person. So it's a big thank you to Jess, great pianist, and now a expedition scientist. And our samples are winging their way back as we speak. We're going to be performing the post-BDC in the DLR, and then hopefully I'll be able to present some of the data from the study. And thank you for your time.
All right, we have time for one quick question, if anyone has any.
Perfect.
Got lucky. Good. Thank you, Mark. Okay, next up we have Professor Alexander Schucker from the Ludwig Maximilians University of Munich.
Johnson. Yeah, thank you very much. I realized, to be very honest, that this session could have been renamed to Redox session or something like that, because I will, this is the pointer over there. It's also about stress, yeah, and the monitoring of inflammation with biomarkers in the air that you breathe. And I would like to introduce a little bit the topic. So what we breathe out, every breath we not take, but take out of our body, this is the composition of everything that we breathe out here. For this gentleman here, big one, and us, and more or less a mouse, maybe exactly the same. So this is a composition, but when you sum up this percentages, there's 0.1% missing. And this is the interesting part. Yeah, there are, of course, one noble gas here, argon, the other noble gases, and then trace gases, and we talked about sulfur already. So it's really interesting. We have not arranged this in that way, so it's really a surprise to me as well. And there are volatile organic compounds that are really of interest to monitor things that are happening in our body, so to say, to bring in a broad context. Wait a second. And these, in humans, we have about 3,000 that are identified that we breathe out every time, maybe not always the same amount, but they are interestingly linked to diseases or conditions, if I may say so. So airway infections, probably metabolic changes. And by the way, if you drink alcohol and you are arrested by police, you know, this is exactly what the volatile organic compound, yeah. And also there have been studies investigating the cancer, you know, small cell cancer, whether there will be in a print for breath print for that. So these biomarkers are really a hot topic because it's non-invasive. The ways you can analyze, you know, this golden standard is mass spectrometry. It's normally the size of a wash machine or something like that. And there are all smaller tools that I will explain in this presentation a little bit more. Generally speaking, not only the air has a kind of finger footprint or breath print, also bacteria. Look, this is like a barcode. When you see here the different masses, culture of Escherichia coli that all of us carry in and on us is producing. And another, the medical doctors in the audience know this, Pseudomonas aeruginosa, very dangerous germ for disease in intensive care, etc. So they have a completely different barcode. So to say, different masses show different, how to say, species or the strains of the bacteria. And here it comes to what we can measure in patients, for instance, on the air that you exhale. And this was an interesting study we did about 10 years ago where we investigated the breath, so of intensive care patients. You know, this is, for instance, one with a real failure of the lung after lung transplantation. And we have intermediate, so ICU sick patients and healthy ones. And when you compare these, the propionaldehyde, which is also a marker of oxidative stress, you can see in the healthy it's very low, and there's a logarithmic scale, and it goes up when you have patients who undergo very severe inflammatory damage and stress on oxidative stress in the lung. It would, just to bridge it, that not only artificially, because it's very interesting to have these non-invasive tools in space as well. And there's a lot of interesting applications, and I will show you a few examples, you know, how you can use this tool. You just breathe or just analyze the breadth of bacteria somewhere to understand whether, what pathogens do we have on board, what are the compounds. We can do immune monitoring, probably recognize specific pattern indicating a specific germ strain or a certain threat of contamination. And there I will show you some data from the ISS on Earth, almost the same. So to understand, specific finger, foot, breath prints, so to say, of what we exhale could be very interesting, and I show you also some examples. And here more applications done with the ENOS technology. So let's get it started. This is, for instance, a device that you can commercially buy, but has been modified for an ISS study I show you later. So there are sensor arrays. It's not measuring a compound like a mass spectrometer. It's more a kind of pattern. It's pretty simple. You breathe through a device on these kind of metal antennax filled tubes, and they are blown over a sensor, and then you do with an AI which has been trained before the analysis. So it's 2 years work, 3 years work, and so on. But it's a nice slide to summarize this up. And this is what it is. This is a principle. So any compound induces a certain conductance here on the metal oxide detector change of conductancy here on the detector, and what you measure is a different the micro ohm value, so to say, and there's a pattern. And these are, for instance, 10 detectors, and every detector shows a different slope of these changes in relation to the amount and the composition of volatile organic compounds. So it's really a fingerprint that you analyze. It's quite easy to use, low running costs, and you can put it more or less point of care near the patient's bed or near the astronaut's sleeping bag even. The reason why we started this project with the Russian colleagues on the station, you can see here the mirror, the whole contamination, the biofilm issue. This is very well known, I think, in the community. So because there's no circulation and there are these wet spots everywhere, so it's a little better on the ISS, but people say this was the reason why the mirror was discontinued one day, maybe, I don't know. And when you look, and this is the study that was being conducted with this ENOS, which is now in our lab, where you have different defined contaminated spots that were brought to space and that were analyzed for feasibility, as well as bacterial contaminations on the surfaces itself, when the different places were analyzed, just to, as I mentioned before, to smell the germs, so to say. And this was the result, and it was quite impressive that they could identify the different, strains of the germs that were spotted there in a quite good manner. So it was identifiable which one it was at the end. So quite promising. So we thought, okay, we can do that. And but unfortunately, the war came and this was suspended. But we have taken advantage of the hardware and of the idea to analyze what we plan to analyze there on the crew. And we did a study when COVID kicked off to compare or to discriminate the COVID from the non-COVID patients with the ENOS. So with exactly that one that was built for the station. So the sensor adapted version. And you can see here, these were our patient numbers and patient clientele, so to say. This is a sick one. You can see there's a CT scan from the lung, really a typical pattern of a COVID patient, and there's a healthy one, uninfected. And we was just recently published this, and I show you how we did that. So for instance, and by chance, we had a chance, this was our first patient number 1 in our study, not on COVID, of course, and he was a physician. He was so willing to participate. He said, wow, this is the future. And he was so willing to even to give the rights to take a picture and to publish that one. And you can see here just exemplarily the pattern of the ENOS data that are generated. You see the different slopes of the different sensors, which show a different pattern in the uninfected or the infected patient. And after all, with the analysis at the end of the long, journey with 3 different teams analyzing with 3 different ways to analyze it, I'll show you this in a second. We found out, you can see here, these are all single data sets, that especially the sensor 9 and the sensor 2 shows the best results, as you can see here with the false positive rates, which are very low at the sensor 9. And we measured the upper respiratory and the lower respiratory tract by the exhalation period. This was triggered through CO2. So both were the best sensors, but also sensor 2. And in the sum, this gave the result at the end. And this is here the approaches that we have used, different AI approaches. And at the end of the day, The misclassification error was very little. So the highest that we had was less than 20%, which is very well in acceptable range, but even better misclassification errors were found. And at the end of the day, it was comparable to PCR analysis, and it was much better to the point of care screening. Yeah, gladly COVID is over, so we don't need it anymore, but it's good to know that we have a tool maybe to identify it earlier. And here I want to come already to the end. I thought it should be a short presentation also to give you an idea only. So where we are now, I mean, there are different enoses as we all know, and this is one that was designed for space, or changed for space. We could see already a proof of concept that it works under microgravity, if I may say so. We have just finished a week ago a clinical study, another one in 800 patients. 500 of those were cardiac surgery patients, where we took the breath print, the breath biopsy, so to say, before they went into surgery, to find out whether there's a predictive value on the outcome. So we can certify the patients before they go into the surgery to find out this one needs more attention than the other. And this is we just finished, and there are a lot of controlled patients and COPD patients and lung transplant patients included. So all in all, this is to be seen how valuable these data can also be translated to astronaut screening one day before they go on mission. Good to know whether they have an infection, somewhere in the lung we don't know and that we don't see. A remote location is a very interesting spot as well, when you can think about a small device which is operating automatically. Also the AI behind could be interesting. And as well, of course, when you go beyond Earth's vicinity, far out. But as you can see with the colors, here we are, the yellowish. This is where we are right now. Maybe we have to invest a little bit more brain and thoughts how to realize this maybe for the future. And with that, I would like to close. And these are all the valuable partners which made this possible. And I have really to thank all the patients. And it was a very difficult study to say. In the COVID days, it was really difficult to run that. But these were the companies as well involved in the 3 analyst model. And of course, my team I want to thank very much, and especially the support from DLR in this matter. So yeah, let's look for further translations of this technology. And for now, I thank you very much. And I forgot how this is spelled. I say thank you in Polish as well. Thank you.
Yes, any questions, please, Rodrigo? Yeah.
No, this way. Whatever.
Thank you. Super interesting approach, Alexander. My question is how reliable this breathing method is. For example, if you eat something, does it influence? Do you exercise, that is influence? Do you have to really standardize how you breathe in the apparatus? Or
it's interesting, yes, and both yes and no. Yeah, we could identify if we tested it as well when you have a cup of coffee before. This gives a special signature, or we found out when you ate a pizza before. So this is also true. On the other hand, it was quite robust. Also, to our surprise, I have to admit, we were very, very skeptical at the beginning, because we had these patients, different body weights, different ages, sexes, different. Although they were just admitted to hospital, this was the entry point. So they were all sick, but maybe not deadly sick, but not healthy, So, but this was also a range of quality of disease. But it worked. It was really to our surprise that the pattern, and this was something that I wanted to inject in our discussion before. And there was a paper which came out just a little bit before, a review article on different study on investigating different approaches and finding out what is maybe the better way to identify something, infection in the lung, for instance. And apparently the pattern makes a difference. It's not the single biomarker in this sense, one peak like you may see in the mass spectrometer. Oh, this is it, yeah? No, it's more. It's really the print of the air that you exhale that makes a difference. And to our surprise, all 3 analysts who looked at that in different models and with no intent, just analyzed the data without knowing to which group that was so clear, yeah, that this breath print has. So the data that we may have not seen or that we cannot see in itself gives as a signal this result that is stronger than the confounding factors as that we have anticipated at the beginning. Yeah, really to our surprise. There's another one. I can jump over there. Oh, no. I hope this answered your question. Yeah, you want to go? Yeah. Yeah. Not yet, not. But it will be interesting. By the way, we did also during parabolic flight. There was an experiment. You could really see was interesting. But this one n of 1. Yeah. Please,
Thank you. Very nice presentation. I have a related question. I was thinking about the atmosphere that in the closed environment of the ISS or spacecraft is different than in the normal life. So is what we breathe will make a difference on what we exhale. If this makes it maybe difficult to compare what you observe on Earth and what you observe on the spacecraft.
Yes, there are two elements in your question. The one is the atmosphere itself, which is quite similar. So to say, as you know, there's 1 bar and the oxygen, maybe a little bit more CO2 on the ISS. But in general, it was the idea to have the same atmosphere like on Earth on the sea level, roughly. Yeah. However, I agree with you entirely that the environmental air that you breathe is part of what you rebreathe, of course. Yeah, and this is also the reason why there should be also control, that you have to run a sample that should be taken, from the air that you breathe, yeah, to calibrate for big differences. But nonetheless, we did this as well, but this was not important to the data. They were in themselves so strong that you, with and without this noise of the environmental air that you breathe, it was not a big difference. But I agree, this has to be a control everywhere. When you sample breath air, you have to sample a kind of ambient air as well. Yeah. Okay.
Go for it. One more quick. Oh,
Yeah, sorry, I can run as well. Sorry, I was not sure.
Well, if you don't mind, I would come back to the first talk. Actually, we saw a lot of space analogs here. So, but in fact, they are not really the analogs. And in the first talk, it was really nice because you said that the nutrition is important because it's important for recovery, along with sleep, of course. So that's the normal things we all forget about it. But if we really say, okay, we want to use our space analogs, especially here on the ground, of course, to mimic the situation there, then at least factors like nutrition and sleep, we should standardize to the situation we have in space. And if you look at the bed rest studies, it's very standardized, perfect nutrition, very, very standardized. If you go to Concordia and so on, it's not the space food we have out there. So if we really want to get closer with these space analogs, I think we should standardize much more, especially these factors which are important for regeneration, because otherwise we will really face situations out there we couldn't test on Earth just because of these simple things. So I just wanted to add that.
Jonathan, I think there's something for the final discussion, right? So I agree. Yeah.
Okay, thank you, Alexander. Okay, our final speaker is Martina Maggioni from Charité Universität Medicine Berlin. Martina will talk to us about one of her projects at Concordia.
So, first of all, thank you for inviting me today to present this project, the preliminary results. I'm Martina Maggioni. I work at the Institute of Physiology, so I am a physiologist, as many of you in this room. And I work at the Center for Space Medicine and Extreme Environment at the Charité University of Medicine. But I would like to point out also that I'm an active member of the Charité Center for Global Health. Why I'm saying this? Because I'm I think it's quite relevant to underline that after about a decade of work in space medicine, we develop specific technologies and sensors that for astronauts, first of all, that were implemented on the International Space Station, but they are now implemented in the broad spectrum of global health, especially climate change and health in sub-Saharan Africa. So the same sensor that we develop for astronauts can be very useful to assess the impact of heat, extreme heat, on the poorest people on the world, so subsistence farmers in sub-Saharan Africa, to develop adaptation strategies. And I think this translational example is quite relevant for the world community. Anyway, today we are going to talk about circadian rhythm and behavioral response in a specific extreme environment. Just a little background about circadian rhythm. We have an endogenous clock that is controlling and regulating many body functions. And the most important timekeeper, the Zeitgeber, is the sunlight. There are other cues, but the sunlight until now has been recognized as one of the most relevant. So for this reason, the many disturbances that we can encounter also during daily life, like for example, daily light saving times or light pollution or shift work, which is a kind of very well known in the medicine field, the conditions, they are impacting this internal endogenous clock, and they are impacting in several ways the general health and the human physical and cognitive performance. For this reason, it's relevant also for astronauts to look into this. And Concordia, as already described previously, is a wonderful analog. First of all, Concordia is at high altitude, so as an external factor we have in addition chronic hypobaric hypoxia. Furthermore, it's extremely isolated because it's far away, more than 600 kilometers from any other living beings, and the average annual temperature are about minus 55. There are, however, two different features that I would like to highlight today. First of all, we have a kind of two seasons at Concordia. During the summer, the Antarctic summer, the station is very crowded. We have the summer crew preparing the station for the winter rover and the incoming winter rover crew, the outcoming winter rover crew. So during this period, they experience about 4 months without sunset, the so-called polar days. On the other side, during winter, the Antarctic winter, there are only maximum of 12 people in the station, and they are experienced during this period the so-called polar nights. So about 4 months of total darkness, and temperature can reach about minus 80. So for this reason, Concordia is been recognized as a high fidelity analog for isolation and confinement, replicating from this point of view many features of long-term space mission. So goals of the Cardicortex project, which I'm presenting today, were, first of all, to assess circadian rhythm alteration during overwintering, which is around 13 months, because the crew arrive at Concordia in different time points. So in average, they stay around 13 months. And I would like to stress again that Concordia is not only isolated, but is also the most remote and coldest place on Earth. The ISS can be evacuated in 24 hours. During winter, no one can reach Concordia. So the crew must be able to face any kind of emergency and to be completely autonomous. The second goal was to investigate the additional effect of high altitude. So the additional effect of hypobaric hypoxia on circadian rhythm, of course, but also the additional effect to isolation and confinement, which are two different things. One is to be isolated very far away from other human beings or living beings, and confinement means also that you are in a very narrow place during winter. And finally, we want to analyze the interplay between core body temperature, which is recognized as a marker for circadian rhythm, and cardiovascular autonomic regulation. This is always in the, as my colleagues previously pointed out, to try to implement an integrative approach. So the method, these are the preliminary results of the Winter Rover 22. We decide to implement a set of wearables to perform a non-invasive monitoring, to allow the crew to perform their daily task, and to get some information about the real-life settings at Concordia. And the project itself ran over 2 following missions. So I'm now presenting only preliminary data of the first one. And what we use, they are research-grade wearables. We measure core body temperature non-invasively, electrocardiography with very high resolution, 1000 Hertz, blood oxygen saturation for 2 consecutive nights. This session was performed once a month over the complete winter over period. In addition, our participant crew were on actigraphy watch continuously, meaning 24 hours a day for all the 13 months. Here, I would like just to give you an overview of the device we select. And again, I'm pointing out this one is the sensor to measure core body temperature. That was a different one. This is the new, let's say, upgraded version that was in use on the ISS circadian rhythm project. And this is now mounted in this headband that allowed very, very freedom during the day to do everything, especially also at Concordia. We had to face many challenges, as already reported, to make possible to wear this different device. In addition, to control for possible confounding factors, every month after the end of the session of 36 hours and more recording, we perform a constant work rate exercise test on a bike ergometer, a medical device that we delivered at Concordia. And again, referring to the previous talk, this was also a challenge to deliver a medical bike ergometry at Concordia, and now it's there. For the future project can use the same device and the ergometer. So getting to the preliminary results, these are about 10 men, so the crew of 22, and we have here the individual value. And then fitted the curve. This is the mean, and you see here in the background the different light periods, okay? So changing from December, when it's summer, until the end of the winter over and coming again the summer in Antarctica. And you see a significant decrease on step per day during the overwintering. On the other side, the duration of sleep per night during the dark phase increases significantly in the group. So furthermore, we observe wake-up time. You see here very strange line because some subjects drop out or started later, so you have some individual value, but I think it's important to see also that there is individual variability. And we saw a shift of the wake-up time of about 1 hour. And also, to assess circadian rhythm, we use cosinus analysis. So we look at the cycle of 24-hour core body temperature, looking at the peak and the nadir of core body temperature. You know, core body temperature is changing in a very narrow range because we are an endothermic organism. So core body temperature can change about 0.5 degrees. But we have a fixed period during the day because the circadian rhythm. And at Concordia, during the dark phase, we observe a shift in this minimum during the, let's say, night, early morning. So this pose in favor of observing some shift related to the different photo periods. So we talk about sunlight being one of the most important or the most important sightkeeper. But to provide you a bit more details, I decided today to show you the long-term medical survey results. This is a bunch of psychometric scale and also the Lake Louise scoring system, so for acute mountain sickness that already was mentioned, to provide you more details about the adaptation and what was changing during this first winter over. You see here 2 panels, November and December, because we, as I said, the crew arrive in different time point. And at the beginning, this is a centrally organized by ESA survey. At the beginning, the Lake Louise scoring is done every day. And you see at the beginning a higher score, which indicates more symptoms like headache, like sleep disturbances, dizziness. But however, the value were decreasing during the first week. During the 12 months of overwintering, the scale was done once per month. And you see here the average value of the crew, but also individual value. And you see that the data remain in this black point, approximately the same at the end of the first week, meaning they were acclimatized. However, if you look at the individuals, you have a magenta, orange, and green person here that react completely different. And that's something that we are going to investigate, why in this subject was so different. The most important symptoms that was reported were sleep disturbances. And this is in line with what we saw about the circadian rhythm before. Another scale that is part of the long-term medical survey is the so-called positive and negative affect schedule. This has been also criticized because a self-report questionnaire, but I think it's important also to look at how the perception of the subject, how he's feeling in that situation. And you see here, I grouped the negative affect and the positive affect. As for the negative, it was done once per month. There are no big variation during the overwintering, apart from 2 subjects. Whereas for the positive, there is a very large inter-individual variation. And there were subjects with a very low score all during the period, so meaning less positive effect. The last one, which is interesting to see, is the POMS, so profile of mood states. And we have here different period. The first one is in February 22, when the overwinter start. Let's say the crew arrive around November and December of the year before, and there is a period of adaptation, and then they remain alone and operate the station directly, the winter over crew, in February. And you have here June, which is called midwinter, so complete darkness. Then you have October, which is at the end of the overwintering. And the one I call the last is the very last measurement. So meaning that the distance between October and last is maybe a few weeks, 1-2 weeks, because depending if they were leaving on November or end of October. Anyway, the tension, anger, and fatigue and confusion, you see they are very low. This is a measure of psychological distress. The value were very low, but they were increasing. And they didn't show any relationship with the photo period, for example. So maybe also the duration of the mission is affecting the mood state. On the other side, vigor was very high, vigor, and decrease over the period. So probably, in this case, we have more an effect of the mission and the duration and the stress by staying at the Antarctic station. So to summarize, yes, circadian rhythm are influenced by sunlight. However, there are several exogenous stimuli that can affect circadian rhythm, and we observe in the preliminary results of this subsample, because we have a second winter over to be analyzed, that there were changes during polar nights. However, we have to consider the other cues, as I mentioned, so for example, social routine and stress. And this is a diagram of the station. And this is the sleep quarter, let's say. So during summer, the station is quite crowded, and the crew has to share the room with other colleagues that they don't know, for example. And this can cause stress from one side. From other side, can be also for some people very positive because they have more interaction. In winter, everyone has his own room, and so privacy is more available. But on the other side, there is a feeling of loneliness sometimes. It depends on many other factors. So to provide you an outlook, the next step regarding this study are to purify the CBT, core body temperature, rhythm, for exogenous components, like using activity data or heart rate data. And I'm requesting already, I request already, to get more details about the individual's specific tasks at Concordia. Because if you are the doctor, the medical doctor responsible for the entire crew, or you are an engineer, of course, your social cues are completely different, your activity tasks are different, and this will affect circadian rhythm. Furthermore, I also requested already, I discussed just today with Angelique, more details about the environment at Concordia, so ambient temperature, humidity, because the air is very dry, and this affected also sensor, but also the skin. They had many rash and they had many problems. So I need to have core data to better understand my analysis and what I collect. And also the use of possible blue light or lamp that can affect also the circadian rhythm. With this, I would like to thank you for your attention. And I thank, of course, all the supporting institutions. The ESA research medical doctor is Hannes Agson was the medical doctor of the first campaign in 2022 in our project. He did a wonderful job, and also the crew of both campaigns, because they were so kind. We collect almost 98% of the data we were supposed to collect. So thank you very much again, and I'm done with this.
Thank you, Martina. We have used up all of our time, and I've stolen some of your coffee break. So maybe if you have like a question for Martina and would like to speak to her privately afterwards, apart from that, the rest of you are free to go. So thank you to our speakers and to all the questions in the audience. So yeah, thank you. We'll see you in about 25 minutes. Thanks. Thank you.
Thank you very much.